A rare autoinflammatory disease diagnosis can be overwhelming. That's why we created periodicfevers.com for your patients and their families. There's clear, concise disease information and meaningful advice to help during important life moments. Support at every step shouldn't be rare.
Hello and welcome back. Thank you for joining, and we're delighted to be able to open the formal proceedings of Press 2020 with an excellent session ahead of us. So this is one of our flagship sessions, as many of you know, 2 talks to review the last year, and what a year it has been. One of the talks focusing on the clinical science and the other on more basic science. And I think you'll see a fantastic complementarity in those. At the end of the 2 talks, don't run away from your screen, because all will be revealed at that time about the 2 winners from the Young Investigator meeting, which has just finished. And I'm sworn to secrecy until that time, telling you who the winners are. Today's session has 2 wonderful speakers. I'm privileged to know and work with both. We're going to begin with the clinical science from Professor Ramanan, Bristol, UK. And then we're going to hear the more basic science this year from Pete Nigrovich from Boston. So let me now turn to introducing Ram as we know him. Ram is a consultant pediatric rheumatologist at the Bristol Royal Hospital for Children and the Royal National Hospital for Rheumatic Diseases in Bath, UK. He is the Joint Lead for Research, Division of Women and Children at the Bristol Royal Hospital for Children, Professor of Paediatric Rheumatology at the University of Bristol, and co-chair of the UK's National Clinical Studies Group. Ram, thank you so much for joining us to give your year in review for Press 2020, the clinical science.
Thank you very much, Lucy. It's an absolute pleasure and privilege to be doing this talk of year in review, clinical science. As you say, it has been a very challenging and an interesting year. These are my disclosures. So a few general comments on what I've done and how I've structured this talk. For those of you who might remember, not so long ago when we were all face-to-face, the last press meeting was actually twinned with EULAR. So I got a 15-month window to address rather than the usual 12-month window. So what are the principles I took in deciding how I will structure this talk? Firstly, I structured this talk for the person who's going to be practicing on their own, probably in a setting which is not as well resourced. And I wanted to address manuscripts or papers which I think would change practice globally, not just in centers with multidisciplinary teams or with huge amounts of colleagues or resources. Of course, we've all been impacted by the pandemic, but I'm not going to address the COVID manuscripts, which are of significant importance because we have a whole press session which is dedicated to COVID. Now, whenever you do a talk like this, where you're trying to address manuscripts published over the last 12 months, there's a huge amount of manuscripts, significant quality, but I had to make some omissions. All omissions are my own, all of them inadvertent. And I profusely apologize in advance if I've missed any of your work, which you think is practice changing. I've just tried to choose what is probably most relevant for the widest proportion of people. I'd like to acknowledge 3 people who helped me in this process, reviewed the papers I've chosen. Ilaria Macora, who works in Florence, Italy. Bill Renton, who spent time with us and is now in Melbourne, Australia. Gronya Murray, who is from Melbourne and is now with us in Bristol. So to launch straight in no particular order, the first paper is a condition which probably has a significant impact across Europe, North America, and Asia. And lupus is something we've really not conquered. We still have the vast majority of our children being managed on steroids and disease-modifying drugs. But the world of biologics, which has significantly revolutionized JIA, has not impacted JSLE as much. So this is the first biological manuscript, really, which addresses the impact in SLE in children. It's a randomized placebo-controlled trial looking at the safety and efficacy. If you're expecting me to show bar charts and complex diagrams, you'd be disappointed. My key messages are going to be very simple. So this was a placebo-controlled randomized trial. And at week 52, the SLE response index was not significant in the belimumab compared to the placebo. There are some caveats in this study. Now, the children with lupus which most worry us are the ones with renal lupus or the ones with neuropsychiatric lupus. Now, these patients were excluded from this study, so we'd never know if the proportion of patients who are in greatest need of new medications would have benefited from this. So the bottom line is a very modest benefit, and in my opinion, unlikely to change practice on Monday morning when you're confronted with a new lupus patient who's refractory to the current standard of care. Next, the two conditions, if you ask any pediatric rheumatologist, which trouble them most are systemic onset JIA and SLE. So we've talked about SLE, we'll talk about systemic onset JIA. We have this fascinating approach we've all been aware of for a few years, but at last is published in a reasonable cohort, which is the Utrecht approach of using anakinra as their first choice agent immediately after non-steroidal anti-inflammatory drugs. So what did they find? They looked at 42 patients, and the beauty of this study, they were all managed in a single center, so all the biases were equal for all the patients. They were followed up for a mean of 5.5 years, and they were all treated with anakinra after non-steroidal anti-inflammatory drug before steroids or any other disease-modifying or biologic treatment. And what they found was the median time to inactive disease in this group was 33 days. And at a year, more than 2/3 had inactive disease, and half of them were inactive off medication, which really is the holy grail of our management. At 5 years, 96% had inactive disease, and more than 2/3 were inactive off medication. So really some startling results, but what we will need to see is if this can be replicated in a multi-center larger cohort, and whether the health economics will bear. Because if we can use anakinra only for a few weeks or a few months, it might actually even be cost effective to go first in with a biologic. So really practice changing, in my opinion, if you have access to anakinra. Worryingly, we found that having conquered significant amount of the manifestations of SJIA, over the last couple of years, we were hearing reports of SJIA-associated lung disease. So two cohorts addressed this fairly in quick succession, one by Alexei Grom and colleagues, and the other is by Vivian Saper. And both of them address the same thing, and I'm going to merge the message from both. They looked at 61 patients. Unfortunately, the interstitial lung disease in this cohort of SJIA patients in the biological era, treated with IL-1 and IL-6, is only a 5-year survival of 42%. So this complication seems to have a high mortality just when we thought we had controlled systemic JIA. And exposure to IL-1 and IL-6 was the key factor in 2/3 of these patients. The two key risk factors that this manuscript identifies is trisomy 21 and an early age of systemic onset JIA onset. And the pathology is what we used to think in the 80s, we saw a few patients, was a pulmonary alveolar proteinosis, or what we called as a lipoid pneumonia. So the key message, interstitial lung disease seems to be a rare complication, but with significant morbidity and mortality in SJIA patients treated with IL-1 and IL-6. If you do the bronchoalveolar lavage of these patients, if you work in a setting which you have access to these, Alexei Grom and colleagues have shown that you see high levels of IL-18, CXCL9, and 10. And the lung tissue certainly shows upregulated type 2 interferon and T-cell activated networks. The next is a series of manuscripts. And you're going to say, Ram, you said you'd be basic, but you're not talking about JAK kinase inhibitors and conditions which may not be relevant in the developing countries. But let me put your mind to rest. This is a fascinating paper because the last few years, every time I come to press, I always go back learning about a new syndrome which I think might fit one of the patients I have in my clinic, but I haven't made a diagnosis. So what colleagues in this paper in the New England Journal of Medicine show is that this child, which is beautifully characterized in this paper for those who want to read it, first presents at a few weeks of age to an intensive care unit with what looks like sepsis with an intraventricular hemorrhage, gets managed with supportive care, gets discharged, readmitted again with hydrocephalus, another episode which looks like sepsis. They then diagnosed that this patient has USP18 or ubiquitin-specific peptidase 18 deficiency, which they manage with a JAK kinase inhibitor. Now, but for this is a child who would have died in a few months of age. So what is the message for you from this complex genetic paper? That this is a new syndrome, which is characterized by perinatal onset intracranial hemorrhage. So if you see a newborn child who has a sepsis-like process, low platelets, intracranial hemorrhage, calcification, and liver dysfunction, consider this syndrome. And JAK kinase inhibitor may just help save this child in what would otherwise be a fatal condition. And this leads us to a whole host of conditions we all know for the last 5-10 years, type 1 interferonopathies. We're not going to have randomized controlled trials in this condition because they're very rare. And what do you do in the interim when you have these children with severe illness? You can't use other medications. So these are a few manuscripts. The efficacy of JAK kinase inhibition in haploinsufficiency of A20. Now, I'm not going to address haploinsufficiency of A20, but to put it in a nutshell, if you have a child who presents like Behçet's, but this is the autoinflammatory equivalent of a Behçet's syndrome, which can be refractory to standard therapies, and again, JAK kinase inhibitors might just help. This is a fascinating paper from the NEJM again, which looks at another JAK inhibitor, tofacitinib, at the efficacy in cutaneous sarcoidosis and how molecular analysis might help in why it works. And the primary autoinflammatory condition which we first identified was Aicardi-Gouttier with its intracranial calcifications, with its cutaneous lesions. And this paper is only a few days, if not even a week old. It's in September 2020 in New England Journal of Medicine. And what the authors beautifully show is in Aicardi-Gouttier, the devastating developmental impact, the cutaneous manifestations, which can be quite serious, is significantly resolved. But for me, more than the cutaneous manifestations, what is absolutely satisfying is to see some progression of developmental skills, change in developmental skills. For any of us who have seen Aicardi-Gouttier, it can be very sad to see the inexorable decline in the developmental skills of these children. So moving from really fancy autoinflammatory conditions, which I know access to doing the genetic test is not always possible. Most of us who are practicing pediatricians or pediatric rheumatologists or pediatricians with a special interest know that between 1/3 to half of our clinics are usually populated by children with mechanical joint pain. They haven't got an inflammatory problem, but the load and the impact is significant. And what is fascinating about this paper, which stems from United Kingdom, and I know these colleagues, is a multidisciplinary intervention strategy looking at joint hypermobility. We know that joint hypermobility, particularly in certain ethnicities, can be as high as 25 to 40%. So what these colleagues did was in a single center, randomize individuals with benign joint hypermobility to either a multidisciplinary intervention program, which would have an occupational therapist, a physiotherapist, and if required, a clinical psychologist. And half of them to just standard of care, which would usually be advice, information, and escalation to therapy only if required. Fascinatingly, they found no difference in outcomes between these two groups. Now, before I read this paper, my presumption, my prior bias would be that if you had a child with benign joint hypermobility, they needed a multidisciplinary team. They needed a long session with the physiotherapist, a long session with an occupational therapist. Perhaps in reading this paper, in looking at this controlled trial, it's apparent to us a bit of time and resources might be enough. It's a low-cost intervention, which is as effective. And I think this is an important message in settings with limited resources, where not everybody works in multidisciplinary teams, not everyone has access to dedicated occupational therapists, not everyone has access to dedicated physiotherapists, this study is reassuring that standard of care is probably a good start before you move to specialized intervention programs. We've known for 2 decades now that if you speak to an adult rheumatologist with a rheumatic disease, DEXA scan is an integral part of the follow-up of these adults. And then there are clear guidelines, whether it's ACR or EULAR, on what the DEXA scan score is and what you intervene with. Whereas in children, we always knew, while the DEXA scan might show something, we didn't know what the value of intervention would be. And this trial from Madeline Rooney and colleagues in UK was looking at the role of bisphosphonates in glucocorticoid-induced osteopenia, because that really is the major osteopenia that we face because of the iatrogenic use of steroids. So what they did in this randomized controlled trial, which has a placebo arm, which is very reassuring, and I like it as a trialist, is on a 1:1 basis, the children who were on steroids for a rheumatic disease, any rheumatic disease, lupus, JDM, or JIA, would be allocated to placebo, alpha-calcidol, or ricidronate. So they had 77 children in the placebo arm, 71 in the alpha-calcidol arm, and 69 in the ricidronate, which is an oral bisphosphonate arm. The primary outcome was lumbar spine bone mineral density Z scores, age-adjusted. So they did show for the lumbar spine a significant difference between placebo and alpha-calcidol, placebo and ricidronate. Not so much for the total body between alpha-calcidol and placebo. Certainly, if you just were to look at bone mineral density, being on a bisphosphonate made a significant difference. Now, prophylactic bisphosphonates may be helpful, but I use the word maybe because what we really want to know is if you followed them up for 10, 15 years, do the children on ricidronate get less fractures than those who did not get ricidronate? Now, that is a long-term study. But here and now, it certainly shows that oral bisphosphonates are helpful in children who are on long-term steroids with rheumatic diseases. Again, complex conditions, but it's very important because both FDA and EMA have now approved monoclonal gamma interferon antibody. Some of you in the audience are going to say, Ram, we don't treat primary hemophagocytic lymphohistiocytosis, hematologists manage it. But I'm going to tell you why that's important. So in this open label phase 2-3 study, they included 34 patients, 24 of them previously treated and 7 untreated. And they showed that 2/3 of both groups had a significant response and were able to proceed to BMT when normally the mortality in this group is very high. There is an ongoing study in SJIA patients with secondary hemophagocytic syndrome. So it's going to be fascinating for our patients with MAS, which has significant morbidity and mortality. Although I wouldn't go into the world of basic science, I do this because I don't think my colleague Peter is going to cover this. But one of the questions we always address when we see a child with systemic JIA is which of these children is going to get MAS and who's not? Who do I need to follow carefully and who do I don't? How do I know who's at risk? And this paper, again, from Alexey Grom and Grant Schulert, addresses it with some fascinating insights. They looked at IL-18, CXCL9, and S100 in about 40 systemic JIA patients. And they showed that the children who go on to develop MAS have a very high levels of IL-18, offering the tantalizing prospect that perhaps IL-18 at diagnosis might predict who's going to develop MAS or is at risk of developing MAS. A few papers again on autoinflammatory syndrome and their treatment. Now, FMF is the most common periodic fever syndrome, and there are some patients who are colchicine resistant FMF. And this study shows that in FMF patients who are refractory to colchicine, canakinumab is effective, and there is a sustained response for more than 1.5 years in this open label study. When you use biologics in children with SJIA or JIA, we want to know when can we stop, when can we wean, and is stopping better than weaning? And this study by Pierre Cartier and colleagues, which again is fairly recent, a few weeks ago, addresses this exact question. So they look at SJIA patients on canakinumab who have had complete response, and then look at doing 2 types of dose reduction, either increasing the interval or reducing the dose. And what they are showing is that it is possible to do both, and that you can wean the dose or increase the interval. Now, what I would have liked to see is a third option of can you stop the drug once you've achieved clinical remission, but they went for just weaning the dose in terms of the absolute dose given or increasing the interval. But that still offers the prospect of being able to reduce the dose in this very expensive medication. Tocilizumab, we know that some children with JIA uveitis are refractory to both methotrexate and to anti-TNF agents such as adalimumab. And this phase 2 open label study looked at exactly these patients. And they found that a third of these patients had a 2-step response in the anterior chamber cell count, which is an important one and the key measure, if you like ACR30 for JIA. And a further 3 patients had a 1-step response. So about half of the patients who are refractory to methotrexate and anti-TNF may have some sort of response to tocilizumab. And all 4 patients with cystoid macular edema in this study did get better, which we know is tocilizumab is a very good drug for cystoid macular edema. Obviously, this study is part of my own, and that's the conflict of interest in case you didn't notice. Now, most of us are trying to manage in a very challenging pandemic time, the health economics of how can we give the biologics our patients need while keeping the costs down. And there are 2 manuscripts which talk about the explosion of biosimilars and our apprehensions of switching, or our patients' apprehensions or the parents' apprehensions. And this paper, even though it's a review, looks at the negative perceptions. So if we as physicians have a negative perception in the absence of evidence, we will translate that into a failure. And it's important for us to be aware of this and be positive about biosimilars, which will have a significant impact in cost reduction. We did, again, a small thematic analysis looking at detailed profiling of patients on their views on biosimilar switching in JIA. Most frequent concerns were practical in nature rather than theoretical, and most anxieties could be resolved by reassurance from the treating team. I finished with this. Now, most of what I reviewed told me that the trials we do in pediatric rheumatology are still not what's needed. And this group of clinicians working together with FDA and EMA have tried to come up. The fact is, when you see a JIA patient on Monday morning, you want to know, is the new drug better than a biologic, not if the new drug's better than placebo or if it's better than methotrexate. So we need to move away from withdrawal designs and placebo-controlled designs to doing head-to-head studies, for which we need to work together with industry, academia, our patient and parent population. Thank you very much.
Thank you very much, Ram, for that great talk, covering a huge amount, but also very clear. I think there was really something there for all of us to take away. And so now we turn to the year in review of basic science and how it relates to pediatric rheumatology. And we are delighted to be welcoming Peter Nigrovich to the Press 2020 to give us his view of the year that has passed. Peter is a dual-trained pediatric and adult rheumatologist based in Boston, and his lab focuses on inflammatory arthritis and immune mechanisms related to arthritis. From early next year, he will take up the role as chief of the Division of Allergy, Immunology, Rheumatology, and Dermatology at Boston Children's Hospital, and will become the Saud Chair in Pediatrics at Harvard Medical School. Peter, the floor is yours for your year in review of Basic Science, Press 2020.
Lucy, thank you very much for the invitation. Ram's a hard act to follow. That was a very clinically relevant talk. So I'm looking forward to talking to you about what happened in basic science and translational science this year. My disclosures are here. So the way I approached this talk was to ask what basic and translational studies most changed the way we should think about rheumatology, including pediatric rheumatology. Mostly, COVID didn't make the cut, although I'll mention one COVID paper briefly. And again, just like Ram mentioned, it's just too large a field, and I apologize in advance to the many authors whose work I either didn't recognize or couldn't include for space. And I have a number of colleagues listed here who provided very helpful guidance on recommendations for papers. So I'll do this in 4 parts: arthritis, lupus, autoinflammation, and what I consider the future in terms of genetics. So the best arthritis papers this year, I thought, were on the question of what's in a joint. So the very basic question of what kind of tissue are we dealing with? This paper was particularly striking to me. This is a paper published in Nature in June and looked at the fibroblasts in the joints. And so what these authors did was to do single-cell RNA sequencing of 35,000 non-leukocytes, so CD45 negative cells, sorted from RA and OA synovium. And what they then observed was that there were a number of different populations here using a standard UMAP dimensionality reduction strategy, but that most of the fibroblasts were in this overlapping group where they were distinguished by expression of the Phi1 protein, which is a cell-cell interaction protein involved in sublining cells, and lining cells expressing lubricin, also called proteoglycan 4. So pursuing this observation that these two populations were close together, they thought of them as a continuum. And so using transcriptional methods, they identified a transcriptional continuum between the sublining cells on one end and the lining cells on the other, and noted that there was a fairly smooth continuum between them, suggesting that there's something deriving this the evolution of cells from one to the other. So what is that? And here they made a move which I thought was really striking. So they just simply looked at the tissues for the expression of these two proteins. So in particular, they looked here at a synovial tissue stained with immunofluorescence, yellow here being lubricin, the Phi1 being green, and then the vessels here outlined in red. And what they saw was that it looked sort of greenish around blood vessels and yellow the further you got away. And indeed, when they plotted that out from distance to the nearest vessel, these are closer to the vessel and these are further from the vessel, turns out gene expression, or protein expression actually, by immunofluorescence of the proteins varied in a very direct way as a function of distance from vessel. So is that just coincidence, or is that really the vessels doing something? To answer that question, they went in vitro, and they used a so-called synovial micromass system where you can generate in culture a little tissue that looks like a synovium. And if you include just fibroblasts, you get on this little organoid, a lining layer and a few sublining type cells. And those look by gene expression like lining cells. But if you then include blood vessels, the blood vessel cells sort of organize themselves into these little tubes inside the micromass. And then fibroblasts gather on the outside of them. And it turns out now you can see the gene expression signature of that other population, which are the sublining cells. So it looks like the vessels themselves are actually driving this organizational strategy. And so to find out what the mechanism of that was, they looked at things made by the endothelium that also had receptors on the fibroblasts and latched onto this protein called Notch3 on the fibroblasts. And in fact, when they then blocked Notch3, either using a Notch3 knockout mouse or an anti-Notch3 antibody, they were able to show that reduced arthritis in this standard IgG inflammatory arthritis model called serum transfer arthritis. So this paper introduced what we might call a vasocentric model of the synovial architecture, as well as a new arthritis target. So of course, there's not just fibroblasts, but also macrophages. And in this paper published in Nature in August of last year, they generated a mouse that expressed a fluorescent tag protein called TD-tomato under the control of transcription of a chemokine receptor for fractalkine. And somewhat surprisingly, it wasn't a mixture of fibroblasts and macrophages that they saw. What they saw was that the macrophages all sort of lined up like sentinels along the inside of the joint, where this is the lining of the joint, this is the synovial fluid, and this is the articular cartilage. Looking by EM, they saw the same thing. So here are the macrophages. This is pseudo-colored as red, and green are the fibroblasts. And the macrophages were on the outside, connected to each other by tight junctions. And they could see something similar in human tissues. So it looks like the macrophages are playing a kind of a lining role as well. Is that relevant? To understand that, they generated a mouse in which they could specifically delete those lining macrophages using a diphtheria toxin system, and then showed in the arthritis context that the joint became leaky to MRI contrast. That is, these macrophages were preserving the integrity of the joint space and protecting it from things coming in from the blood. And so if they then deleted these cells and tried arthritis, they showed that the arthritis was actually worse. So here we have a new view of macrophages being the good guys in the joint, not just the pro-inflammatory cells. And in fact, a beautiful paper published subsequently in Nature Medicine evaluated that further using synovial tissue biopsy samples in humans from either healthy humans or patients with new onset RA or resistant RA or patients in remission. And they found that the macrophage population, and we won't bother what the axes are here, were just different between these groups. And when they sorted these cells and studied them, they found that in remission, these macrophages made a pro-resolving mediator called resolvin D1, a pro-resolving lipid. You can see that here. So again, picture is macrophages as good guys. So what did that all mean? I think that in terms of fibroblasts, we had previously been thinking about lining versus sublining as their organizational structure, and now we're thinking about organizing around vessels, which is really an interesting paradigm. In terms of macrophages, we thought of them as the bad guys of arthritis, but now we can think of them as sentinels protecting the joint space and also exhibiting pro-resolving functions, although certainly some macrophages also can cause inflammation. There was also a very interesting paper published in the New England Journal about RNA identification of prime cells predicting rheumatoid arthritis flares, published out of New York City. This was a technical tour de force, really. They took 4 patients who would agree to do weekly assessments of their disease activity by rapid-3. They also were in the clinic monthly. And then every week they would poke their fingers and give you 3 drops of blood, which they would put in a little tube and so that you can isolate the total RNA. And so then in that context, they could see, as the patient flared by the rapid-3 score, what happened in terms of the RNA-seq signature. This is what the data actually look like. So again, here's patient in remission according to study day. This is the rapid-3 score rises during the flare, and the yellow dots are prednisone calming things down. And then when they looked at the transcriptional signature, they saw things that you might not be surprised to see in a patient who flared. So different genes which were involved, for example, in myeloid signaling, so in neutrophils. So that, I think, was not particularly surprising. But what's more surprising and more interesting was when they looked before the onset of flare. So here they looked at what they called antecedent clusters, so clusters of genes that went up and down before the flare started. And what they saw was 2 weeks before the flare, here week -2, they saw a spike in gene signature, and then a different spike, right, just 1 week before. And we looked at these signatures. This was a number of different leukocytes. They focused particularly on B cells. And this one was one that they didn't expect to see. So the genes expressed here included tissue genes, bone genes, cartilage genes. So that looked to them like mesenchymal cells. And in fact, when they took these signatures, these antecedent 2 and antecedent 3 signatures, and lined them up to transcriptional signatures from different populations within the joint, B cells, fibroblasts, monocytes, and T cells, they saw that the AC2 cluster lined up with B cells and the AC3 cluster lined up with sublining fibroblasts, so some of the more angry or inflammatory fibroblasts. And when they went backwards and looked in their gene signature for the genes that are associated with sublining fibroblasts, they were able to again see that kind of spike 1 week before disease started. So are there really circulating fibroblasts? To address that question, they didn't have cells, remember, from all their serially monitored patients. They just had RNA. So they went to prevalent RA patients and age and sex matched controls and got a lot of blood and went to look by flow cytometry and found that there was a population of podoplanin positive cells which was a fibroblast marker which they could see. And these are relatively rare, about 1 in a million, but they were there. And when they did RNA-seq on these, they saw that there was a transcriptional signature that which was consistent with a fibroblast, and it looked like the ones from fibroblasts in the joint. And they call these prime cells, meaning pre-inflammatory mesenchymal cells. So far so good. They proposed the following model, that B cells come up early before the flare. They recruit fibroblasts, which then go to the joint and become inflammatory. So what the authors are proposing here is a very interesting hypothesis that B cells, when they're activated, recruit prime cells And these then go to the inflamed synovium to help trigger inflammatory arthritis. And I think that's a major claim, and data don't quite get entirely there. I'd highlight that there were relatively few of these cells, at least in prevalent RA patients, where at this frequency, 3 drops of blood, like the investigators obtained, would have on average only about 0.2 cells. So I wonder whether 0.2 cells can drive a whole blood signature, although it is, of course, possible that there are many more cells in the pre-flare phase. So these data just need validation. Remember that potoplanin is a general fibroblast inflammatory marker. So I don't think we know for sure from these data yet that these prime cells are really synovial fibroblasts. And finally, I'd highlight that there are many links in the chain here that are still just hypothetical. So for example, there's no evidence that B cells and prime cells interact, or that prime cells go to the joint, or that they have any pro-inflammatory role in arthritis. So overall, I think the paper is really interesting, but the hypothesis still needs confirmation. Moving on to lupus. So not surprisingly, a very prominent paper in this field was from Virginia Pasquale's group with Jacques Banchero, studying single-cell RNA-seq on patients with 33 childhood-onset lupus patients and healthy donors. They looked, again, with all these many 200,000 plus cells, they were able to construct a map of the different kind of lineages that were present. And they showed that subset frequency varied across with lupus compared to healthy patients. And in other studies subsequently, that adults and children were fairly similar. They looked to then see which cells are contributing to this interferon signature that we see in the blood. And here yellow indicates interferon signature, and the columns are the different lineages. And they saw that the interferon signature is coming from 8 different lineages, not every lineage, and most probably from the monocytes. Interestingly, they were not able to identify the cells actually making interferon. Does this subsetting of cells tell you what kinds of patients there are? Are there patient subsets? So maybe. So here they looked in yellow at the abundance of the different cell types, and were able to cluster cells into 6 patients into 6 subtypes, though those subtypes didn't really correlate with clinical features indexed here. So maybe or maybe not really clear. So this paper shows that blood distributions differ between lupus and healthy, that the interferon signature rises from many lineages, especially monocytes, although the interferon source is not clear. And they looked at that childhood and adult onset diseases are fairly similar. Most importantly, it's an amazing resource going forward. Moving on to autoinflammation. Here, there was a very satisfying paper that finally sort of put the nail in the coffin of the idea that, or confirmed the idea that Mediterranean fever was selected by plague. If you're looking in Turkey, in the Turkish population, they were able to see extended haplotypes here by these linked disequilibrium plots surrounding 2 prominent MEFV-associated variants. And using mathematical modeling, they were able to track M694V down back to 197 generations to a single donor. Again, 726, about 163 generations, and showed how it was selected by 2 plagues that occurred in the relevant area over this time. Experimentally, they were able to show that the mutant MEFV bound less well to a Yersinia transcription factor called YOP-M, that FMF PBMCs made more IL-1 beta when exposed to Yersinia, and that mice bearing these variants were relatively protected from Yersinia compared to mice having other variants. Another study from the NIH looked at PFAPA, and here things get a little complicated in the autoinflammatory space. So we think of this sometimes as an autoinflammatory disease, maybe a polygenic one. Here they had 230 patients. That's too small to do a GWAS. So they tested 6 SNPs associated with Behçet's and recurrent aphthous stomatitis, as well as HLA associations. And in fact, they did find that these genes lined up. So there was very shared associations, even corrected for multiple comparisons. And also, interestingly, that there were multiple HLA-1 and 2 associations. They propose a new way of thinking about these diseases called BSDs, or Behçet syndrome spectrum disorders, with recurrent stomatitis, PFAP, and Behçet's on the same spectrum. So I'm less certain about this. I would say that they only tested 6 SNPs, and many diseases share 6 SNPs. So I think it's interesting, but not definitive. I'd point out that the HLA associations are mostly different. My PFAPA patients certainly respond differently than my patients from these other diseases. But I do think that overall, there's compelling evidence there's antigen-dependent T-cell activation going on. Another disease in which we often think of as autoinflammatory is systemic JIA. Here in this paper, Lauren Henderson took new onset and chronic patients and looked at their T regulatory and T effector cell populations by a number of different cellular strategies. She was able to find that in new onset patients, T regulatory cells exhibited more IL-17, an IL-1 driven cytokine in T cells, and that could be seen also in their transcriptional signature. By contrast, in chronic patients, that signature switched to the effector cells, although we didn't see IL-17 protein signal. Interestingly, when the patients got treated with anakinra early, that IL-17 signature went away. So do these data prove the biphasic hypothesis? That is, there's an autoinflammatory phase transition to an autoimmune phase? No, it doesn't, but I think the data are supportive. It does mean that we should pay attention to T cells more. And another reason to think about this are these two papers looking at T cell mediated inflammasome-dependent IL-1 beta production. So very important papers, which I can't go into the details, but let me tell you the punchline. So normally we think about IL-1 beta coming from the inflammasome. TLR stimulation leads to precursor production. The inflammasome production leads to caspase 1, and IL-1 beta is produced. However, in these papers, they show that the same process can be driven by T cells in an antigen-dependent way, entirely independent of the inflammasome. So a T cell activated via its T cell receptor makes TNF that drives precursor production, and then expresses Fas ligand, which activates caspase 8, which is another way to cleave IL-1 beta. iNKT cells, which recognize lipid antigens via CD1, can also do that. That is, you can get to IL-1 beta in an inflammasome-independent way via caspase 8. And I think this makes us think differently about immune diseases. So when we think about autoimmunity or an allergy, these are antigen-dependent diseases versus autoinflammation, which I think we should think of now as antigen-independent inflammation. And I think we should be strict about this, although there's a third axis of immunodeficiency. So we proposed a definition of autoinflammatory diseases in which these are diseases in which pathogenic inflammation arises primarily through antigen-independent hyperactivation of immune pathways. Not all our diseases are that way, and we have to be careful. So I think in general, we've been hammering any disease that has fever or IL-1 with the term autoinflammatory, but I think we should be careful with that hammer. Finally, let's look at genetics in the clinic. This is where COVID appears for the first time in this, or the only time in this talk. So in this paper published in JAMA, the group was able to show that young men with very severe COVID had defects in the X-linked gene TLR7, which recognizes SARS-CoV-2 as a single-stranded RNA via its endosomal location. So these are diseases, so that implicates TLR7 and downstream interferon production in morbidity. Taking that approach to its extreme, this paper published in Nature in July looked at primary immunodeficiencies of adult onset in over 800 patients, looking both at known genes and in different other kinds of sort of known high penetrant mutations and other ones, such as rare variants, non-coding variants, or common variants. These are relevant to us because of the autoimmunity was also prevalent in many of these patients. Essentially, what they found here is about 10% did have documentable rare gene variants, some of which were unexpected in their variants, some which would have been hard to pick up by normal whole genome sequencing, and some had unexpected presentations. They were also able to prioritize some rare variants. And the take-home point here is that whole genome sequencing looks like a very interesting approach for immunologic disease. Finally, how do we bring this to the clinic in a more practical approach? This work from Shomari Chowdhury's group took advantage of the fact that RA, lupus, spondyloarthritis, psoriatic arthritis, and gout all have genetic signatures, either HLA or lack thereof, or non-HLA SNPs. And they asked, can this approach be used to help diagnosis? They generated what they called a G-prob, a sort of a score, a genetic score reflecting pre-test probability, and combined then genotype and synovitis, in patients with synovitis, to prioritize diagnoses in individual patients. So this patient has more lupus, this patient more likely to have spondyloarthritis, this patient more likely to have gout. And then in a validation cohort of over 200 patients, they showed that whereas doctors were correct about 65% of the time, the GPROP score wasn't bad, about 53% of the time, and it was able to aid in prioritization, excluding one diagnosis in all patients, and getting the right diagnosis in the top 3 in about 87%. And they were able to demonstrate quantitatively that there was a quantitative addition between having genetic data with clinical diagnosis and making it better. It does add measurably to the clinical score. And I think it shows how genetic data, hopefully in future, will inform clinical decisions in real time. So what did we learn? We learned that fibroblasts orient on vessels, that macrophages can be good guys, and maybe that prime cells are interesting, although I think there's a lot of work to be done there. We learned that lupus has a complex cellular landscape and is a fertile ground to seek cell subtypes. We learned that FMF variants of MEFE were likely selected by plague, and that we have to be a little careful about what's really autoinflammatory and what's maybe not so clearly so. And finally, genetics is a tool. So with that, I'll thank you for your attention. And for those of you interested in joining us in Boston, postdoctoral positions are available. Thank you.
Thank you, Peter, for a really excellent talk, for bringing to our attention so much of what's happened in basic and translational science in the last year, and explaining it so beautifully for a wide audience. In fact, there were some interesting links, I think, between both of these fantastic talks that you've just heard. So we heard about IL-17 in systemic GIA, and this may actually underlie the early response to anakinra. Don't forget, you can go back and review these talks and look in detail at all of the papers, because the material will be available to you both through the Congress and afterwards if you've registered. In addition, after these fantastic talks, you can post questions on the meeting system. So go on there and ask any questions you may have. But don't go away now, because very excitingly, the time has now come to reveal this year's 2020 Young Investigator Prize winners. There are 2 of them, and that meeting, the Young Investigator meeting, finished only a few hours ago. So hot off the press, their names will now appear on your screen. And following that, you will be able to hear both of these prize-winning short talks in this session.
Good afternoon, I'm Roberta Caorsi, and I'm going to present you a study about pediatric recurrent pericarditis treated with interleukin-1 blockers. Here are my disclosure. The aim of our study was to evaluate in a cohort of patients with recurrent pericarditis and treated with interleukin-1 blockers the appropriateness of first-line treatments, the long-term efficacy of different interleukin-1 blockers, the number of patients able to achieve a drug-free remission, and the presence of factor predictors of drug-free remission. In order to do that, we performed a multi-center retrospective study, and we collected pediatric patients with recurrent pericarditis, both idiopathic and post-pericardiotomy, treated with interleukin-1 blockers. The analyzed relapse rate was used to evaluate the efficacy of different interleukin-1 blockers, and the bivariate logistic regression analysis was used to identify possible predictors of drug-free remission. We collected 58 patients with a median follow-up of 2.6 years. 57 patients were treated with anakinra and 6 with canakinumab, meaning that 5 patients were treated with both drugs. Here you can see in red the treatment received by the patient in the first month after the beginning of the disease. And as expected, it was mainly represented by NSAIDs and colchicine. While before the beginning of treatment with interleukin-1 blockers, in green, patients were mainly treated with steroids. At last follow-up, in blue, just 9 patients did not receive any drug, while 39 were still in treatment with interleukin-1 blockers. Here you can see the treatment with the first line drugs, and you can see, as you can see, most of the patients that did not present an adequate response both to NSAIDs and colchicine did not receive a proper dosage according to the European Society of Cardiology guideline. On the opposite side, most of the patients treated with steroids were overtreated, and 38 of them displayed a steroid dependence. Here you can see the response to treatment with interleukin-1 blockers, and you can see that there is a significant reduction of the analyzed relapse rate in treatment with anakinra than before treatment, even though we observe an increase in this rate after the reduction of or discontinuation of the drug. Patients treated with canakinumab presented a reduction of the relapse rate than before any treatment, but the 5 of them that received both treatment had an increase of the relapse rate in treatment with canakinumab than in treatment with anakinra. When we compared the 9 patients that could withdraw all treatment with the 26 that couldn't because of the recurrence of the disease, we didn't identify any variable with a statistically significant difference. In the same way, the bivariate logistic regression analysis did not identify any predictors of drug-free remission. So in conclusion, our study confirms the effectiveness of interleukin-1 blockers in pediatric recurrent pericarditis, even though most of the patients require prolonged treatment to maintain clinical remission. In our cohort study, canakinumab turned out to be less effective than anakinra in the control of the disease manifestation, and we observe a high rate of steroid dependence. Finally, the statistical analysis did not identify any predictors of drug-free remission. So in light of these findings, we think that longitudinal study as well as control trial are needed in order to identify the best treatment of patients with recurrent pericarditis in the pediatric age. Thank you very much for your attention.
Good afternoon, ladies and gentlemen. My name is Sjia from the Department of Pediatric Rheumatology of the Erasmus Medical Center. Today, I will give you a brief summary of the results from our study on the effect of immunosuppressive medication on the type 1 interferon signature in an in vitro model. Systemic lupus erythematosus, as you know, is characterized by increased interferon-stimulated genes known as the interferon type 1 signature. This signature has been linked to disease activity in some cross-sectional studies. Furthermore, standard of care drugs like hydroxychloroquine and aspirin have been proposed to inhibit this interferon activation. For this, we wanted to study the role of standard of care drugs on the interferon signature in an in vitro model to better understand potential pathways of interferon activation in vivo. In our study, we induced the interferon pathway by 3 different stimuli in freshly isolated human peripheral blood mononuclear cells. Standard of care drugs were added to cell cultures for 24 hours. After this, cells were harvested for real-time quantitative PCR, and MXA was used as a readout for the interferon type 1 signature. As you may see, all stimuli linked to the different routes were able to induce the interferon signature. The toll-like receptor 7 route induced by imiquimod was completely blocked by the addition of hydroxychloroquine, while mildly downregulated by aspirin, diclofenac, and high dosage of prednisone. Additionally, the toll-like receptor 9 route was also completely blocked by the addition of hydroxychloroquine and mildly downregulated by diclofenac. Lastly, the C-gas route induced by a C-gas agonist was clearly not affected by the addition of hydroxychloroquine. Data from our longitudinal cohort showed that after treatment, patients recover clinically, but the interferon signature does not change in a subset of patients. This indicates the involvement of multiple pathways in interferon induction. Earlier, we showed that hydroxychloroquine was able to inhibit the toll-like receptor 7 and 9 route, and not the C-gas route. This last named route could be an explanation why some patients remain interferon positive after hydroxychloroquine treatment. In a separate study, we show that the effect of hydroxychloroquine was indeed overruled by a CGS agonist, indicating that interferon production is still possible under hydroxychloroquine treatment through another route. We confirmed that this interferon activation is pathway specific by showing the upregulation of phospho-STING only when the CGS agonist was added to the culture system. For this, we conclude that standard of care drugs are able to inhibit the interferon signature. Furthermore, as we show with our hydroxychloroquine experiments, multiple pathways are involved in interferon activation. For this, these results may explain partially the discrepancy between the interferon signature and disease activity in a subset of patients. Thank you for your attention.
Thank you both very much for excellent talks, and may I congratulate you again on being Young Investigator Winners 2020. So you've heard an amazing breadth in this session. I think it really sets the scene for our meeting this year. I'd like to thank all of the speakers, all four of them, for wonderful talks and for educating and interesting us throughout this fantastic session. Don't go too far away. After your quick cup of tea, you will find that there's an exciting and live COVID session. Be there. I think it will be of interest to everyone, and you'll be able to post your questions in real time and ask the speakers all of your questions about COVID in children. Following that, lots of other exciting sessions towards the end of this afternoon and today. Take your pick, have a look through the program, and enjoy, learn, and engage. Thanks for joining us.